Hello students, welcome to my channel with yet another learning video. These days we are discussing some important questions and this is the second video of the same series, right? So if you have not watched my initial video, please do watch it. Now here again I have picked a question and it is asking what are recombinant DNA vaccines? Recombinant DNA vaccines, how are they prepared and what are they? Remember the concepts that were told to you in chapter number 11 that would be written here. Means we'll be picking the gene of interest of our choice. We'll be mixing it with the plasmid DNA and then we will introduce in the host to get a protein. So that protein will act as recombinant DNA vaccine, and once it will be inserted or it is taken, vaccines, they have a general phenomena that they include the antigen in a slow form. I mean a little amount of antigen is being inserted in the body which elicits or evokes the immune system of an organism and the immune system starts preparing antibodies. Same funda will be applied here also, but only the recombinant DNA vaccine will be prepared by the process of recombination and transformation. So recombination means recombinant DNA will be prepared, it will be introduced in the host, that protein that will be produced will be used as recombinant DNA vaccine. They are asking us to give two examples, and examples are hepatitis B vaccine and bird flu DNA vaccine. Remember, we can use bacteria and yeast for the expression in the case of vaccines. The second part of the question was asking the advantages here, because it was written discuss their advantages. So see here, important thing is, which is common to every vaccine, that we give the antigen in the killed or slightly active form, which is known as attenuated, and it evokes the immune system. Another thing with DNA recombinant vaccines is that they are highly pure. Specific means they will target only specific cells of the body, and that specific cells will be the cells of the immune system. Right? So you should know the name of two recombinant DNA vaccines to answer this question, and your advantages should be concrete, right? All these terms should definitely be included. Now let's move on to next one. This question I have picked and along with the answer so that you can pick up the keywords. Drugs like LSD, barbiturates, amphetamines are used as medicines to help patients with mental illness. However, excessive doses and abusive usage are harmful. Enumerate the major adverse effects of such drugs in humans. So you need to include few symptoms like harmful effects of LSD and barbiturates are anxiety, shakiness means shivering in the body, every time it feels like vomiting, which is known as nausea, sweating, loss of mind control, reckless behavior, vandalism, violence are also being seen. Lack of interest in personal hygiene, because the person is not conscious. How is it? The person is going to look after the hygiene. Fluctuations in the weight and in the appetite, withdrawal, isolation, not merging with others, and when the person will not say his or her inner to the another person, automatically the person will undergo depression, tiredness, aggressive behavior will be there. Social problems will also be there because the person is not interacting, right? Withdrawal symptoms can be severe and life-threatening. Means if the person is on all these drugs since long and immediately he is being you stop suddenly, then their symptoms could be devastating. Excessive doses of drugs may lead to coma or even death may occur due to respiratory failure, heart failure or cerebral hemorrhage. So whenever withdrawal has to be done, it has to be done in certain amount of a dosage. Like it's not like a person is taking continuously and one fine day you stop. So the body will not respond immediately and it will not get settled, rather any of these may happen. So if such type of question comes, you pick up some of the effects of your own choice and please do involve that in your answer. I hope this is understood. Let's move on. This is question number 3. Given below are the pairs of pathogens and the diseases caused by them. Which one of these is not a matching pair and why? So this question has been taken from chapter 8. This question 2 was also from chapter 8, right? So virus causes common cold, it is absolutely correct. Salmonella typhi causes typhoid, this is also correct. Plasmodium causes malaria, that is also correct. So the option number C is the one which is not correct, right? Why is it not correct? Because the causal organism of filariasis is Vaccaria bancrofti, okay? So the causal organism here is being given wrong, that's why it is wrong. So you need to pick up option C and you need to write the causal organism again, that Vaccaria bancrofti causes the filariasis, which is a fatal disease of the lymph nodes. This is another question, students, and it is based on HIV, right? So they have given up a concept map and we need to fill this one and we need to fill this one. So it's starting with retrovirus. Virus infects the normal cell, and with the help of retrovirus only we got to know that they are talking about HIV, because only one retrovirus is there in your syllabus. So there is one diagram which is given in the NCERT. They have made the flowchart out of that only. So it is asking after reverse transcription what is formed. So in this block you are going to write viral DNA is produced by which enzyme? Reverse transcriptase. Okay, so if such question come, you need to write like this, and here in the blank 2 you will be writing new, because after this see what is happening. Viral DNA incorporates in the host genome. So after incorporation what is being done? New viral RNA is produced by reverse transcriptase. Okay, so see here viral DNA was produced, or here viral RNA has been produced. So this is the key term, viral DNA, viral RNA, and if you notice in both the cases reverse transcriptase has to be mentioned. Now why is the virus retrovirus? Because retrovirus are the viruses that contain DNA and they show the process of terminism or reversal of central dogma. Reversal of central dogma has to be written as a key word. Now you know that the central dogma includes the flow of information from DNA to RNA to proteins, right? But the viruses that have RNA as a genetic material, they first from DNA and then further the information flows. So this is also known as terminism or reversal of central dogma. Next question is very different. Can the infected cell survive while viruses are being replicated and released? So they are asking about macrophages and helper T cells. Do they survive? So they survive. The answer is yes, but it is only for a small period of time. And this is the reason that a person affected by AIDS, they have less number of macrophages and helper T cells in their body, because they are alive for a while, but being infected, they are being killed by the body, right? I hope this is understood. Let's move on. This is the question of chapter number 10, and it is asking why is distillation required for producing certain alcoholic drinks. So distillation is being required depending upon the amount of alcoholic content you want in the hard drink, right? For example, in your NCERT only they have mentioned that wine and beer, they fall in the category of without distillation, whereas whiskey, brandy, rum, they fall in the category of distillated hard drinks. This is important. So distillation makes the thing pure. The more the distillation will be done, the purest would be the thing, and the more will be the alcoholic content. So this line here which is written is very, very important. Thus I have picked this answer for you. Distillation increases the alcoholic content in the alcoholic drinks. Remember, alcoholic drinks, they can also come in exam by the name of hard drinks. Okay, so if hard drinks word is given to you, that means they're talking about alcoholic drinks. This is the key word. So if they give you two examples and they say that this is prepared by the help of distillation and drink B is prepared by not utilizing or by non-distillation process, which drink has more alcoholic content? So remember, always pick the drink that has undergone distillation process, right? So in this case, you can give the example of whiskey, brandy, rum, even vodka comes in the distillation drink. Whereas wine and beer, they have less alcoholic content in them because they are non-distilled hard drinks. See you in next video. Please do share these video with your friends so that they can also get the help. Wishing you all the very best. Stay connected to my channel. I have lot more questions for your A's. Thank you.